Hi, and welcome to my OCR A-level biology revision session with me, Christine. So today's lesson, I want to look at respiratory substrates. So let's just take a step back and remind ourselves about aerobic respiration. So aerobic respiration is where the glucose in the cytoplasm is broken down into pyruvate through the process of glycolysis. Pyruvate is then converted into acetyl coenzyme A through the link reaction, and then the Krebs cycle occurs, and all of the coenzymes are going to go to the cristae for oxidative phosphorylation to occur, which gives us a high yield of ATP molecules. So glucose is our respiratory substrate of choice. It is the one which would be broken down very easily. And through the process of breaking it down, what happens is oxygen is required as the last electron acceptor of the ETC, and therefore when we look at the what's known as the respiratory quotient, we can calculate this by figuring out how much oxygen is going to be consumed and how much carbon dioxide is being produced. So we know that the link reaction, decarboxylation, occurs, and we know that through the Krebs cycle, decarboxylation also occurs. So that's where our carbon dioxide is going to be produced, and at the cristae, as the last electron acceptor of the ETC, that is where that oxygen consumption needs to occur. So we know that for one glucose molecule, we need 6 oxygen molecules, and that will produce 6 carbon dioxide molecules and also 6 water molecules. So carbohydrates therefore have what's known as the respiratory quotient of 1. 6 carbon dioxide produced, but 6 oxygens needed to be consumed. Well, if the carbohydrates are not available, or there are other substances available, they can also be utilised as a respiratory substrate. So we need to look at where could they come from. Now we know our energy storage could be in the form of glycogen. The glycogen can be hydrolyzed down to glucose. Well, glycogen is a polysaccharide. It's still a carbohydrate. So what's our other storage of energy then? And that comes in the form of our lipids. So our lipid molecules, we know, can be hydrolyzed down into glycerol molecules and fatty acid chains. Well, it's important that we note a glycerol molecule is a 3-carbon molecule, and therefore it can be converted into pyruvate. So if it's converted into pyruvate, that can then be utilized in the link reaction and the Krebs cycle. With the fatty acid chains, what would happen with them is they would actually be broken down into many acetyl groups. So it can be broken down into lots of 2-carbon groups, and that can enter at the end of the link reaction to go to the Krebs cycle. So as long as the coenzyme A is available to pick up the acetyl group and deliver it to the Krebs cycle, it can then go and be utilized in the production of our ATP. But remember, it's important to note that the lipids need to be hydrolyzed down before they can enter into the pathway. Also, the acetyl group needs to be joined with the coenzyme A. So it's a bit of a harder process to occur, and therefore when it comes to our lipids, we actually have a respiratory quotient of 0.7. So in this example that I've given you, there were 25 molecules of oxygen consumed, but only 18 molecules of carbon dioxide that were produced. That therefore gave me a respiratory quotient of 0.72. Now we know if the number for respiratory quotient is close to 0.7, that is signifying that lipids are the respiratory substrate of choice. So what happens if there is no glucose, no carbohydrates, no lipids? Well, if the body has got no other reserves to use, it will then utilize the proteins. Now, the proteins are actually going to give us a respiratory quotient of 0.9. And why is that? Well, we need to look at an amino acid structure and remember that our proteins are hydrolyzed into amino acids, and then our amino acids are deaminated in the liver the amine group is removed, and that leaves us with the remaining carbon chain. And that remaining carbon chain can then enter, as it can be converted either to pyruvate, or it could be converted to the acetyl coenzyme A, or it could be converted into any of the intermediates within the Krebs cycle. Now, what that tends to mean is when it is the proteins that are being utilized, the respiratory quotient is 0.9. So in this example, the oxygen consumption is 0.6 and the carbon dioxide produced is 0.54. So whatever they give you in the exam, for example, they could tell you that there's some peas that produce 37 carbon dioxide molecules but consumed 46, which would give you a respiratory quotient of 0.9. If that were the case, what could you conclude from that information? Well, what you could conclude is that protein stores were what the peas were utilizing. If we're looking at the wood lice was RQ of 1, so they were utilizing the carbohydrate stores. And if it is the sunflower seeds and they have a 0.7, they are utilizing their lipid stores. So this is a way they could ask you to apply this knowledge. The other way they could do it is they could give you a picture that looks like this, which is telling you you have a person who is doing some exercise, and it's looking at the change of the power of that person that's exerted during that exercise process. So the first thing I would say when you get a question like this, figure out where's your carbohydrates, where's your proteins, where's your lipids. We know carbohydrates are 1, proteins 0.9, lipids 0.7. You have to memorize that. Now it's about the interpretation. If it is closer to 1, then that organism is utilizing its carbohydrate stores. If it is at 0.9, it is proteins. But what we can quite clearly see is we're not really that close to 1 at the beginning, and we drop down and we never reach the 0.7. So therefore, what we tend to say is when the number is between 0.8 and 0.9, which is normal activity, that's showing that both carbohydrates and lipids are being used. We also have proteins being used, but only those that are in excess. So our amino acids that are in excess are going to be deaminated at the liver. Therefore, those carbon skeletons can be utilized. So the majority of the time when we talk about the normal activity, we would tend to say 0.8 to 0.9 is both the carbohydrates and the lipids. If it were to reach down to the 0.7, we would state that that was the lipids that were being utilized as a respiratory substrate. But what happens if it goes above 1 then? Well, if it goes above 1, what that's telling you is there's more anaerobic respiration occurring. The reason there's more anaerobic respiration occurring is because the intensity of the exercise is much greater. That means that there's not going to be enough oxygen, so they're going to produce lactic acid. Now, lactic acid is an acid, so that's going to affect the pH. Now, the way in which the body responds to that is it will actually increase its carbon dioxide level to stop the blood pH becoming too acidic, as the lactic acid is being transported to the liver to be converted back to the glucose. So it's really important that you note anything above a 1 means that we're mainly utilizing our carbohydrates, but also we're also doing a lot of anaerobic respiration, because if it was aerobic respiration, we should have an RQ of 1. And as we do more intense exercise, we would need that ATP being provided at a faster rate. Therefore, anaerobic respiration would take over until we get the supply of oxygen to the level which will ensure that they are aerobically respiring. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, do share this in my TikTok session if you're with me, because the more students that I can help, the better it will be for you guys when it comes to the exam. If you haven't already done so, please do also check out my revision platform, www.aiqchat.com.